What if I told you that if skin cancer is detected early, patients have a 99% survival rate after 5 years? Except the reality of diagnosing melanoma is that patients are facing unnecessary, long, distressing waiting times before receiving a potentially life-changing diagnosis. Today, melanoma is diagnosed through visual inspection. However, this subjective assessment refers many patients to unnecessary and invasive biopsies, where samples of skin tissue are removed for examination. Each biopsy costs a minimum of $150, and 90% of those biopsies don't even have melanoma. So how can we bridge the gap between emphasizing early detection and ensuring that only the right patients are selected for a biopsy? Our solution is mPatch. mPatch is a microneedle patch that offers a minimally invasive, pain-free screening alternative. The needles are coated in a sponge-like material to absorb fluid from the skin, rich in clinically informative biomarkers. Our patch is applied for 5 minutes next to a suspicious spot, and during this time, skin interstitial fluids is absorbed through the patch. Our preclinical studies have been successful with an accuracy just as good as the standard of care. But we'll be doing this much faster by reducing waiting time from 2 weeks for a referral to 24 hours for a result. We're asking for £1.5 million for our clinical studies in human tissue samples. And with mPatch, we're not just redefining the standard of care, we're empowering healthcare professionals to make timely, informed decisions. Thank you.
Gentlemen, questions?
Start with the regulation again. So you sort of say it's a microneedle array you put next to melanoma. Have you had regulatory advice, or are there any similar products on the market that you can refer to?
So we'll be based in the US market. We are referring to our nearest competitor, which is Demtech. That our device will fall under a Class 2 device, and we'll be following the FDA 510K pathway.
The biomarkers that you've got in the patch, are they identical to the current biomarkers that are used in laboratories?
So we'll be looking at micro RNAs as our biomarkers. Currently, other companies are looking at genetic data, so the complete DNA instead.
And in terms of the analytical technique that you're looking for your biomarkers, is there anything unique about that, matching up your biomarkers that you're looking at versus the analytical technique?
So with other patches, they have a sticker that sticks directly on top of the lesion, as opposed to adjacent to the lesion. We'll be sampling the skin interstitial fluid while they will be looking at the actual histopathology of the debris. So in that sense, we would have different products.
Have you given any consideration to how this would be put alongside some of the new scanning, automated scanning systems that have been developed and deployed now more widely, which are mostly used in the private sector here in Europe, but probably will come into the public sector where you can actually screen people very quickly and effectively on a yearly basis, particularly for younger people, where if you're taking a skin, basically a whole body scan, you're able to detect any changes very quickly and effectively.
Of course. So currently visual inspections are being carried out, and I believe that the latest trend is also AI, but we'll be looking at the molecules themselves within the skin interstitial fluid and being able to give a pretty accurate diagnosis.
So you're envisaging that this would then be used by dermatologists that are using that technology.
That's correct.
Right. Well, thank you very much, Sarah.